Paolo, I lost my mouse for some reason. I think it's charged, but for some, I'm not seeing my. Do you have control of my screen?
No, no, sir.
Let me just see if I can advance it. Huh. Maybe it's a charging issue. I don't know. Let me just plug that in. Do we have 2 minutes? Do we have 2 minutes? 1 minute?
Yes, 3 minutes actually. And
Okay.
I believe your camera is also frozen.
Yeah, you're right. That's so strange. Let me just go make sure no one is using the internet here. I'll be right back, okay?
Okay. Okay. Dr. Richardson, can you hear me? For all the audience who follows our broadcast, in a few minutes, we will have Dr. Richardson connected with us, and he will start his lecture, Epilepsy Network Surgery. Thank you. Thanks to all the audience who follows our broadcast. In a few minutes, we will have Dr. Mark Richardson in his lecture, Epilepsy Network Surgery. Thank you. Dr. Richardson?
Just waiting for the PowerPoint to reboot.
Okay, it's okay. Just to be sure that your microphone is okay, as well as your camera, please.
Sure. I see, it's, there we go.
Okay, your camera, it's okay.
Let me just check one thing here. Let me just see if it saved this work that I just put in. Okay, can you give me just 60 seconds?
That's okay, doctor. No problem. To all the audience who follows our broadcast, we are about to start Dr. Richardson's lecture on epilepsy network surgery. Please stay with us.
All right, I'm ready when you are, Paolo. Sorry about that. Oh, let me switch it for you.
So, Dr. Richards, okay, perfect. It's wonderful. So, thanks to all the audience who follows our broadcast. We have the honor today of having Dr. Mark Richardson. Dr. Richardson is the Director of Functional Neurosurgery at Massachusetts General Hospital Charles A. Pappas, Associate Professor of Neurosciences at Harvard Medical School and visiting Associate Professor of Brain and Cognitive Sciences at MIT. Now, at the IWBNC, Dr. Richardson is going to share his lecture titled Epilepsy Network Surgery. Please write your questions in the Q&A panel. We will read them after the end of Dr. Richardson's intervention. Welcome, Dr. Richardson, and thank you. The microphone is all yours.
Thank you, Paolo, and thanks to everyone for hanging in there. Not sure I've had that happen exactly before the computer crashes, literally right before the televised broadcast, but I'm back. I think I got all the slides there that I hadn't saved. So I'm going to give two talks. The first one is on epilepsy network surgery. And then the second one, for those who are interested, will focus really on DBS for movement disorders, but with broader implications to DBS for psychiatric disease too. I've tried to organize both of these so that I won't talk longer than 40 minutes for sure, and would love to have discussion based on the questions. So please don't hesitate to send those in. We had a lot of good questions when I did this last year. So really appreciate the invitation to come back and speak. All right, so let's talk about epilepsy network surgery. And I'm going to click. This is one that didn't get back in place, so that's okay. We'll do this one first. So let's talk about the difference between a focus-oriented and a network-oriented approach. So in a focus-oriented approach, one traditionally is just thinking about identifying one spot. You know, what's the primary hypothesis, and let's find that one spot that's potentially curative. And a lot of the backdrop for this type of thinking is, for some people, well, epilepsy surgery is really only worth doing if you can cure the person. So we need to find that one spot that we could resect and take it out. Okay, but if you can't find that, then in some cases, with some philosophies, let's say there may be no surgical remedy, or someone might get a VNS, or they might get a thalamic DBS, which might be okay. Thalamic DBS works pretty well. But let's say, yes, the seizure focus is identified. The group says, we know where seizures are coming from. We're going to take it out, or we're going to ablate it. Well, what if that's in the Wernicke's area, or it's somewhere else that's been mapped for language or other eloquent cortex? Well, we can't perform this. So again, no surgical remedy, or we could go to focus-directed RNS. Again, not a bad option in that case. Let's say you can do the resection or ablation. And then the next question, of course, is the patient seizure-free? So if they are, then this pathway has worked great. And in some ways, thinking about this simply is the most efficient and the most reasonable way to proceed. But if the patient's not seizure-free and outcome's not achieved, then it seems to me that it's easier to get stuck in this loop of thinking, well, we must have missed it with our primary hypothesis. Maybe we should have just taken a little more. Maybe we can ablate a little more. We should have made the temporal lobectomy bigger. Let's try it again. Or, well, we did a temporal lobectomy, let's put an RNS. And then you can go back again, try to find the seizure focus. Oh, and here, if the patient's not seizure-free, one could additionally go to non-specific neuromodulation. It might be too difficult for the team, may say. And this is, I think, unfortunately the case a lot of times for a patient. The surgery doesn't work, and so there are no other real options offered, or patient kind of falls off the tracks. So in a network-oriented approach, I think there's a little bit more potential to not get caught up in the idea that that there's only one focus that has to be found, and if we don't find that, it's a failure. We always still want to, if there is one spot in the brain that can be resected and cure someone, we always want to try to attempt that. But in identifying the network, I do think there's more of hypothesis testing, because a network approach requires there to be more than just a primary hypothesis. And in the focus-oriented approach, I do think it's possible for the group or the surgeon to think we've nailed it. I think it's this one thing. There's no backup plan. And so if that doesn't work, the patient can get stuck. So when we think about the seizure network, I think we have to think about what the other alternative hypotheses are, because part of testing the network hypothesis is to think about how the seizure spreads throughout the brain and how the clinical seizure manifests as a result of that activity. And then if we think more along the lines of how can we take the network offline as opposed to what can we take out of the brain that's definitely going to make the person seizure free, then we still have that one option, resection. We can think about ablation. We can think about using those together, and we can think about network specific neuromodulation. So in order for neuromodulation to be network specific, we need to define that network. Excuse me, of course, we can make some assumptions about that. It's essentially the kind of philosophy of anterior nucleus DBS, that we know a circuit and we're going to put the electrodes there. But it's that depending on how that approach is done, it may not be specific to that patient's network. And then if we think of a desired quality of life improvement, there may be some patients who say, I don't want to go through any of this unless I'm going to be seizure free. But I think the majority of patients want a significant improvement in their quality of life. And through multimodal surgery and thinking about all the options and kind of keeping secondary hypotheses kind of at the forefront of our thinking and thinking about how we could address those if we're wrong about the primary hypothesis, that we stand the best chance of improving the patient's quality of life. And that is the ultimate outcome that we want to achieve. But let's say we don't do that in our first round. We have this idea of combining therapy. So sometimes through doing stereo EEG, for instance, we might have pinpointed what we think is the worst part of the seizure network, attempt to take that offline with resection or ablation, but have the backup plan that we understand the network. And if that's not sufficient, then we might do neuromodulation, or we might do some other combination. And then, of course, if we may have to reevaluate the whole hypothesis. It's always a possibility, and we should always be willing to do that. I think the chance of getting it right the second time is greater if we've been really thoughtful about the network the first time. Okay, so this is a statement that's made a lot. Epilepsy is a network disorder, but what are the practical implications of this statement? So here are a couple thoughts. One is that we should think beyond a primary hypothesis. And this is one thing that struck me more and more in conversations about this general philosophy of networks. Secondary and tertiary hypotheses are required, and they should be tested when all the data is not concordant, or at least mostly concordant. This is a potentially kind of bold statement, but if we think about all epilepsy as a network, it could be a very local network, it can be a network as broad as idiopathic generalized epilepsy, that this idea in terms of surgical treatment or surgical candidacy about whether it's focal or non-focal epilepsy is a false dichotomy. We should think about how we can treat all types of epilepsy surgically. One reason we can do that is, in my opinion, because of the advent of closed loop stimulation in the United States. That's the RNS system, responsive neurostimulation. Now the Medtronic Percept device is available. It's not currently unlocked for closed loop DBS, but presumably that will happen at some point and be available for epilepsy. I think when we think about any brain network, we know that function and dysfunction travels through basal ganglia thalamocortical pathways. So having an understanding of the role of subcortical structures in seizures is very important. Obviously, the thalamus being kind of top of our list. So with respect to that, when do we need to understand the role of thalamic activity in seizure propagation? or even recovery from seizure. And how do we know if or when we should include in stereo EEG, and when we should potentially move to a thalamic or thalamocortical neuromodulation strategy for the patient? I do not have all the answers to this, but we'll touch on a few things here. Now, this one is definitely out of order, but hopefully we will find the, here, let me just look at this real quick. It is. Okay. I'm going to come back to that because really we want to be here. So before we talk about IgE, let's talk about this paper. So I think this is a really important paper that really everyone should be aware of. It's the first case series reported on the treatment of regional neocortical epilepsy with corticothalamic stimulation. And in this particular series, this is from the Spectrum Health Group in Grand Rapids. David Burdett's a neurologist there, and Sanjay Patra is the surgeon. So they have taken a very thoughtful approach to modulation of regional epilepsy circuits with thalamocortical stimulation, with the hypothesis that that's the best way to hit the broad circuit. So I've just pulled this directly from their paper because I think it's interesting. So the next slide will show the results, but you can see, and I'm pretty sure in this case series, all the patients had stereo EEG. So there was some data to describe the network. And you can see the diffuse or variety of MRI findings in this population, and a variety of strip electrode placements. So in this case, the two available RNS leads were placed, one in the thalamus intermediate nucleus and one in the cortex at these locations. And you can see these really significant reduction in seizures. And there are significant follow-up in these patients. This is what it looks like just by histogram. And these results are quite good. This is an open label case series, but this is very promising. And this group has continued to think about this and think about implanting different areas of the thalamus based on how the network is defined. For instance, implanting the pulvinar for regional occipital epilepsy, which makes sense. And there's some data available from SEG studies where the pulvinar has been implanted that implicate the thalamus in that type of regional epilepsy. Okay, so here we'll switch to IgE. So again, sticking with the RNS device, and it has been in my practice the default to use that device for neuromodulation in the brain because of its ability to record data, record seizures. And we really believe in the value of that, especially collected over years in patients. Publications from the UCSF group indicate the cyclical nature of seizures that can be determined from these recordings. And to me, it seems very clear at some point, patients will get some feedback from that device in some form or fashion related to seizure forecasting. There's evidence that RNS is useful in medication titration. And so for these patients with IgE that we came across when I was practicing in Pittsburgh, who the first person came to us as someone who did not want VNS, was not able to take Depakote, and really not a great option. There are a lot of patients like this with IgE, and there are a lot of patients also with secondary generalized epilepsy of various types who really don't have a great surgical option. Many of them have failed VNS, or that's not a good option for some reason. So the question is, can generalized epilepsy, can these seizures, these generalized spike wave discharges be detected in the thalamus? And it turns out there's a lot of evidence already in the literature existing that was the case from DBS studies. primarily the group of Velasco and colleagues who had implanted depth electrodes as well as externalized DBS leads and recorded seizure activity from the thalamus. And specifically, they targeted the centromedian thalamus in their work. More recent data from this group in London who used centromedian stimulation to verify that connectivity is essentially with frontal lobe for generalized epilepsy. Again, with externalized DBS leads, then doing open loop DBS with some significant benefit in patients. But again, having this evidence of recorded activity also detected in the centromedian. And in particular, in this cohort, patients with generalized epilepsy did quite well in terms of their reduction in seizures with centromedian stimulation. A couple outliers there. And here's another case series from a Korean group looking at lead location for centromedian DBS stimulation, again, separated by type of generalized epilepsy, and those with idiopathic generalized epilepsy did quite well with little variation. So there's reason to think that this is valuable and appropriate for patients. So we showed this last year. We had a case report for one patient, young woman who responded very well. She has Jovan syndrome, which is eyelid myoclonic absence seizures with eyelid myoclonia. These are the details of her case. And now we finally have compiled the data on the 3 subsequent patients that were implanted in Pittsburgh. So I'll share that with you. We're getting ready to submit that, but thought I would just go ahead and share that. This is the first patient who reported 99% reduction in seizures. We can still see seizures on her RNS, but in terms of how, her thoughts and how they impact her, there's a massive reduction. Oh, and that's in the setting of completely stopping medication, which the patient did of her own accord. These are her detection and stimulation locations. So we're starting to go back and look at the recordings and metadata in a lot more detail. And one thing that we did notice this is a generous screenshot. The electrode's really skiving the back of the centromedian nucleus. So if you look here, based on the color code and where we're detecting and stimulating, it's not in the centromedian nucleus. And we wrote about this in the case report. And it's because the targeting is off a little bit, but this was highly efficacious. So we're exactly sure what to make of that in terms of locations being stimulated. Here's a second patient. Again, a lot of seizures, huge impact on quality of life, 10 ER visits and 6 ICU admissions in the year prior to RNS. Now is reporting 90% or greater seizure reduction. No hospitalizations, et cetera, driving. Okay, here's that person's lead location. So again, just kind of skiving the centromedian here. But we managed to hit it, this one, and to do some detection and stimulation there. What you're going to see as I show each of these is that there's really a lot of variability in where we're recording and stimulating. Here's a third patient. We may have filled in this data since I made this slide before, but 2 generalized tonic-clonic seizures per month, doing very well, better than 75% seizure reduction. Here are her lead locations. Again, in the centromedian here more, we're using this. And again, these are examples of detections. Fourth patient from this series, greater than 90% reduction. Frequent generalized tonic-clonic seizures before, none in that should say past 7 months. Okay, and here are that person's locations. So here it looks like the leads were a little deeper. And so we're using the top contacts, but these are, for the most part, in this intermediate nucleus. So what does this mean in terms of outcomes? So this is a chart that shows each of these patients' long episodes, which are probably the best proxy we have for ictal events or epileptiform activity that's recorded on the RNS device. And patient 1 does not download the data very often. So that is an important caveat of getting the most out of that system, is the patient does need to interact and participate and do the downloading, which this patient does not like to do. And so I think her long episodes may probably look different than this in reality, but nonetheless, this is the data, and she's been one of our best responders. These other patients make more sense. You see the long episodes going down over time, and you see in each case, it takes a few months. These are days on the x-axis. All right, and this is what the demographic and outcome data looks like for these 4 patients, and you can see the Engel scores are pretty good, especially when you can consider that these patients did not have surgical option aside from VNS. And we don't know that these patients wouldn't have done just as well with VNS. But I think one thing we've shown with this case series is that we can expect to detect seizure activity in IgE in the thalamus. And we know we have activity that can be tracked over time. And so this, personally for me, is the option that I recommend first to patients rather than VNS for medically refractory IgE. Okay, let's talk about central median targeting for a minute. So here are our electrode locations from that case series I just showed you. And we have one extra electrode. One of these patients had an implantation, maybe a thalamic, a cortical one also. I think that's included here. There are 5 electrodes on each side. This paper was really instructive for thinking about how to target the central median nucleus. And I've shared this with other people, and I'll just talk a little bit about how it changed the way I target. And clinically, we didn't have a reason to change the targeting, but it made sense to me when this paper came out to try to do a better job of targeting the central median nucleus, which was our original intention, and that we would see whether there's a difference in the outcomes. So I think this is the key image from that paper. And this is an axial view. So what this group did was to develop this MP2 RAGE sequence, and then they do some pretty fancy post-processing, edge contrast enhancing, and essentially kind of made their own atlas for doing this. But in your own center, if your scanner can do the sequence, you can do your own MP2 RAGE. It does not look as nice as this, but you can see the border of the central median nucleus. And I really should have pulled up one of ours to show you that it doesn't look this nice, but it's better than just, for instance, an FGator or a Bravo or MP RAGE sequence. And the key view here, where this is best visualized, is 3 millimeters superior to the posterior commissure. So the current consensus coordinates, the personal consensus coordinates that used for this are plus and minus 8, minus 9 in relation to the ACPC midpoint, and in the 0 plane. And so I put the target there, and then I will scroll the trajectory up, which I just use a standard entry point around the coronal suture. We'll scroll to 3 millimeters above the target plane and assess whether or not the lead is going through the center of this intermediate nucleus to the best of our ability with that particular patient scan. This is something else I got out of that paper, which is microelectrode recording is not that helpful, it seemed to me, was the take-home message in terms of finding this intermediate nucleus. I think it's an open question about the utility potentially of doing evoked potential mapping to determine that you're in a part of the thalamus that is connected to the cortical region that you are hypothesizing or have proven with SEEG is involved in the regional epilepsy, regional seizure network. But we have not done that. And then here's our targeting. It is not perfect every time, but it is certainly these are a better place for the most part within this intermediate nucleus, and the signals are better. And we have done a few patients since being in Boston. And the first two of those patients are also responders. So we're 6 for 6 on response of RNS for IgE. So I think that's it. And I'll just stop there, and we'll take questions, and I'm happy to discuss any aspects of this or anything I didn't cover. I really focused, as you can see, mostly on the thalamocortical question, because this is, it's certainly an area of interest that we think about a lot these days. So I'd be curious if others are implanting, the thalamus in their SEEG cases, and when they're doing that. I'll just make a couple other comments. The way we have started doing that for some patients, it's certainly not routine. The way we use that information is really to assess the latency of the seizure onset in the thalamus compared to that in the cortex. And if we have a network that we think is a regional network, and we're trying to determine whether to implant the thalamus as part of the strategy for RNS, or to do 2 cortical leads, let's say, we determine that based on how quickly the seizure propagates through the thalamus. And there's reason to think in the literature that in IgE, the onset in the thalamus is, in some cases, more or less simultaneous with cortex. In the regionalized epilepsies, we expect the thalamus to lag the cortex, but the idea is that through thalamic stimulation and kind of broad innervation of cortical regions, that that strategy more broadly affects cortex than putting leads into random, not necessarily random, but in 2 spots in the cortex. So even if there's a little bit of lag, if the lag is too much, and we don't have a definition for what that is. But if it's very clear that the thalamus is not part of the primary organization of the seizure, then we don't implant the thalamus in that case.
Well, thank you, Dr. Richardson, for such a wonderful lecture. So we have here some questions from the audience. Alim Bayramov is asking 2 questions. So the first one is, what percentage of people who have surgery for right temporal lobe epilepsy find that it doesn't work and need to have additional surgery?
That's interesting. Well, it depends on average, that the success rate for let's say right temporal lobectomy, which you might expect to have the highest success rate. If the data is concordant, that the success rate could be 90% seizure-free at 1 year. Let's say on average, it's 60 or 70%. But what we do know is even those patients that do really well initially, after 5 years or longer even, can still have a seizure. And so that number on average across temporal lobectomies, drops to just below 50% in terms of patients who are seizure-free. But there are a lot of different factors that it can account for failure after temporal lobe surgery. I'm curious why the question was about right temporal lobectomy.
Okay, thank you. And the second part of this question is, a person was told that even though their surgery was 100% successful, there was a 30 to 40% chance the seizures could return if the person was to come off their medication. Why is that? Thank you beforehand.
That is a great question and something that's probably understudied. So one thing that we don't understand about epilepsy networks when they form is why we can remove part of the network and have someone be seizure-free unless they stop their medication in some case. And the best explanation for that is that even a focal epilepsy can affect neuroplasticity across a broad region. And then if one comes off the medication too soon, or in some cases, someone can't come off the medication, enough of the surrounding brain has been hijacked, essentially through plasticity mechanism, and can generate seizures even when the primary site of organization has been removed. So I think that's one possibility. The other possibility is that in general, we may not always be resecting the area of primary organization sufficiently, and that in the presence of medication, someone can do very well with the remainder of this network there, but without, they can't. And then another possibility is that in general, epilepsy broadly affects the brain once seizures happen and take hold, that it's almost always a more global problem, or a wider network than we realize. Because most patients do have to stay on their medication.
Okay, doctor, thank you. Dr. Camilo Peña is asking, is it possible to treat an epilepsy patient with DBS and also an RNS?
Aha. Well, it's certainly possible. I'm not sure if that, I'm not aware of that having been done, but it's very interesting. So in that case, that is a very interesting suggestion. One reason that you would do that is because currently with both those devices, one is limited by the number of sites it can be implanted. And so you can imagine that you had mapped a network where you wanted to record and or stimulate in more than 2 places. So for instance, it's conceivable to do duty cycle stimulation in the thalamus and have the RNS detecting and simulating in another, let's say, cortical part of the network. But I've not seen that done before. Maybe someone has done that.
Thank you. Dr. Richardson, I might ask you, what is your opinion on machine learning or deep learning in order to improve technology for epilepsy hypothesis testing?
Aha. Well, I think it is potentially extremely useful. The limitation is the quality of the data, however. So if one wants to apply these types of methods, they will only be as successful as the data organization and collection is to begin with. So does that make sense? We spent a lot of time in my lab and practice focusing on the data and focusing on collecting good data. Now, the intracranial data is obviously objective. It is what it is. But it's important to understand the context in which the, let's say, stereo EEG data was recorded. It has to be annotated. For instance, right. So often there's a clinical infrastructure that works, it does its job. But that infrastructure is often not ideal for extracting information for these types of higher level questions. And so making sure the data is correctly organized and annotated as it comes out of the clinical system into a research database is critically important. But I think there's a lot of potential, especially with respect to the stereo EEG data.
Perfect. And continuing with this topic line about technology-assisted surgery, on your experience, is it always necessary to do surgery with the frameless robotic-assisted ROSA robot?
Yeah, well, it's certainly not. Oh, tell me the question again. In what context is it? Could you just repeat that again?
For example, on DBS or for example, bilateral thalamic stimulation, do you always perform this type of surgery that will include eventually deep structures on the brain? Do you always use your ROSA robot?
Aha. Yes, I do. So I have converted essentially all stereotactic procedures to use the robot for a couple of reasons. One, having a single workflow for every stereotactic procedure is helpful. And so we use that for DBS for movement disorders. We use it for stereo EEG, and we use it for RNS. But it's not, obviously it's not necessary. Important thing is to get the electrodes in the right spot. There are multiple ways to do that, but yes, in our practice, it's been the most straightforward to just convert everything to the ROSA.
Thank you. Here we have an interesting question, and for the young surgeons which intend to configure an epilepsy group, is there always necessary or mandatory to include a neuropathologist in the group?
Aha. Gosh, that's a great question. So this is a question related to, for instance, starting an epilepsy surgery program.
Yes.
Is that your interpretation of the question?
Yes, sir.
Okay. It's ideal. Is it necessary? No. I think in my mind, it would be more important to identify people who are candidates for epilepsy surgery and perform the correct operation. I would not let the lack of a neuropathologist hold me up from doing that. Having said that, there are obviously, it's always helpful to know what's on the pathology, especially in the setting of focal cortical dysplasias, because there are some types of focal cortical dysplasia, like type 1s, that can be diffused through the brain. And it's helpful to know when you have one of those, and for instance, that would prevent you from doing repeat resection after resection in someone who really has a widespread problem. For the most part, it doesn't change the treatment plan for the patient.
Okay, doctor, thank you. I have here also another interesting question, and is due to the nature of refractory epilepsy, can you consider that in a future the closed loop DBS could be included as a curative approach? I
do think that's possible. I don't have any evidence any data to support that, but to me, it does make sense that there would be some epilepsy networks that we would be able to develop an understanding of and apply control theory to with electrical stimulation, especially as capabilities of these devices improve, that we would essentially be able to stop someone's seizures. It wouldn't cure the underlying pathology, but I do think there's a potential to make people seizure free with closed loop DBS. Yes, for sure.
Okay, wonderful. Dr. Madriñan is asking, are there any neural restoration, for example, as in Parkinson, for dopaminergic neurons, restoration therapies used for epilepsy treatment?
Short answer is no, not that I'm aware of. However, I think there is currently interest in developing gene therapies for epilepsy. In general, there's a lot of interest now in gene therapy for central nervous system disorders, including those that could be treated with direct delivery to the brain, or spinal cord. So I do think that we'll see gene therapy clinical trials in epilepsy over the next few years. Cell transplantation is an idea that has been proposed for epilepsy. I'm not aware of any new or current ongoing work that would get that sort of thing into the clinic. But I do think there's a lot of reason to believe that some epilepsies will be particularly well suited for gene therapy. Even if you just think about temporal lobe epilepsy, the idea of doing infusing a therapy, whether it's gene therapy or something else in the temporal lobe, is a very safe way to start for someone who is otherwise a candidate for temporal lobectomy. Because if there's a safety or other issue, patient can proceed with their otherwise planned surgery. So that's really an attractive a subtype, to attempt gene therapy for epilepsy in particular.
Perfect. Here is another question. What is the imaging protocol for an epilepsy surgery candidate?
Okay, great question. Typically, we get a 3 Tesla MRI, but I have had this question before. Well, how necessary is that? We don't have a 3T. How often do you find something else? With really good 1.5 Tesla imaging, this can be almost as good. I do think that 3T is better. The key is to get good volumetric scans. So let's say someone has a 1.5 Tesla scanner. The key is to get good volumetric scans, develop protocols, especially with the T2 imaging. So I think the advantage to 3T, and we have a 7T that we don't use for every patient, but we use for some patients. The advantage to the higher magnetic strength is seeing subtle gray matter abnormalities. It could be a focal cortical dysplasia. And same thing with high resolution T2 imaging. I think looking for FCD and potentially seeing differences in the internal structure of the hippocampus are the two, probably the two big things that stand out that you have a better chance of assessing maybe with higher Tesla. But I don't think anything can replace the value of the surgeon reviewing the films on his own. I think that is one of the things that's most likely to reveal a lesion, is the surgeon spending time and reviewing the images on her or his own.
Yeah, it makes sense. But for example, continuing with this topic of imaging studies, is this same protocol included during intraoperative MRI, or do you use some different sequences?
Well, we don't really, at least in my practice, we don't really use the intraoperative. Well, that's not true. So we may use intraoperative MRI to do an anterior nucleus DBS, for instance. So to finish the question, the other parts of imaging are often a PET scan, which can be very helpful. You can also do, you can attempt ictal SPECT. So that's a CT scan, though, that's done with a radio tracer that's injected at the time of seizure. For that to be useful, the best approach is actually to have an injector that automatically injects this as soon as someone notices there's a seizure. We most of the time, we get a MEG scan, magnetoencephalography, which has, I think, variable success in helping us delineate where the seizure focus is. It can often help guide where we put stereo EEG electrodes. 7T is an option at some centers. Some centers can do PET and MRI together, which is helpful to see. I think that covers it. And for then, and related to your question, intraoperatively, we talked about the specific sequences for targeting the central median nucleus. The anterior nucleus can be targeted with relatively standard sequences that we would use for DBS. You can see the anterior nucleus. So in most centers, that doesn't require a specialized scan. It requires just ordering the right one.
Okay, thank you. Is there a case where WADA testing will still be necessary before temporal lobe epilepsy surgery?
Okay, another good question. I don't think so, but I'm very biased. I haven't used WADA testing as part of my practice for really the entire time. We did that still a little bit in Pittsburgh, and then it was replaced by language lateralization by MEG. And at MGH, we do language lateralization with fMRI. So I think that's the main benefit. There are a few publications about one's ability to do a temporal lobectomy in so-called wrong way WADA. So the other thing about the WADA is one can't always take the memory testing results at face value because of the potential for there to be cross flow across hemispheres. So I think in a center where a specialized protocol for WADA is arranged with endovascular surgeon, that's probably where it's most valuable. A standard WADA, I think, is just not necessary.
Okay, wonderful. And I believe we have our last question for this Q&A session. And do you currently conduct any translational research in epilepsy surgery network?
Yeah, well, most of the, yes, we do. We're trying to understand these networks better in relation to the RNS and how the RNS works. And we're in the process of relating the stereo EEG information we have to recorded seizures and stimulations and outcomes with RNS. And so I would say that's the main translational project related to networks in my group.
Well, that's wonderful. So Dr. Richardson, thank you for your wonderful lecture once again. And for all the audience, in a few minutes, Dr. Richardson will start his second lecture of the day called Evolution of DBS, Closed Loop and Connectomics. Stay tuned in this room to watch his lecture. Please, Dr. Richardson, in order for us to continue this lecture, please start your presentation, share your screen, and we will do a brief introduction right now.
Hello, Dr. Richardson.
How are you? Hello, how are you? Sorry, I accidentally stopped sharing.
Don't worry. We are going to wait until 4 o'clock to start with your conference. So we are just going to wait until then to start the conference, your second conference, your second lecture.
Great.
Nice to see you again.
It's so nice to see you too. I'm sorry about the technical difficulties. And then I lost some other slides. I don't know if you noticed the talk was a little short. There were a couple of things that were left out, but I think it worked out okay with the questions.
Yes, it was okay. It was okay. Don't worry about that.
How's everything going with you?
It's okay. It's okay. We are oversolving all the pandemic. We actually have also some elevated cases, however, but the vaccination is going on. So fine, getting over that.
Good. And what's going on with your practice now? What?
I'm actually with the hospital where I did my residency, and in another small hospital, and looking forward for my training and the fellowship in epilepsy. So waiting for all the COVID pandemic to get end.
Right. And then what's your plan from there in terms of, so you're going to do a fellowship?
Yes, I'm waiting for all the COVID pandemic get end, but after that, like start with all the program, which has already started on our hospital, but to get it more get it better.
I see. Good. Well, let me know what I can do to help.
Of course, you will know. Okay, good afternoon for all the audience who stay connected for the second lecture of Dr. Mark Richardson at the 2021 IWBNC. He's going to share his lecture titled Evolution of DBS, Closed Loop and Connectomics. Please write your questions in the Q&A panel. We will read them at the end of Dr. Richardson's intervention. Please continue, Dr. Richardson, and thank you. The microphone is all yours.
Well, thank you. Okay, so thanks everyone for sticking around, and welcome to anyone new who may have joined. We'll switch from epilepsy to DBS for movement disorders and psychiatric disease. Yeah, please do send your questions in. There'll be plenty of time at the end to go over those. So although we're switching diseases, you'll see a kind of similar theme, actually, of understanding the network physiology and why that's important. And so let's just jump right into this question. Not whether we should have closed loop DBS, but why should DBS be closed loop? And there are a few reasons. Even though DBS works pretty well, on average, it doesn't work perfectly in any patient. And in some patients, it doesn't work as well as we expect it to. And that can be because of side effects that aren't explained necessarily by a bad lead location. It can be something specific to the patient's disease, particular symptom that is more intractable for them. Simple things like reducing battery life with the amount of current that is delivered. And Simon Little, who's been a pioneer in closed loop DBS, published a paper a few years ago about less speech symptoms with closed loop DBS. Wasn't clear exactly why that might be the case. It might just be less stimulation all the time. And we don't understand how the basal ganglia exactly process speech, but being able to record the correlates of different behaviors from different brain regions could be very important for how we deliver therapy and DBS and how we can tailor that for individual patients. So I guess a short answer to this question would be, in order to personalize DBS for a specific patient's physiology or symptoms. All right, so we'll talk about the extent to which that's possible. We'll skip the slide. Okay, so this is a schematic that shows my interpretation of the potential evolution of closed loop DBS. And so this is the stage where we are now, which is sensing-enabled DBS. So with the advent of the Medtronic Percept system, one can get some brain recordings. The actual closed loop features are still locked, so this is just sensing-enabled, so everything's manual. In adaptive DBS, DBS system adjusts its stimulation on its own in relation to a trigger. This would kind of be like the RNS, how the RNS works for epilepsy. There's a trigger, the device is either on or off. You can imagine different variations of adaptive DBS in terms of what the signal is exactly, and how it's modulated when it comes and goes. And this type of system, of course, still requires attention and kind of manual adjustment of medication. You can also envision, this is where we really like to be, intelligent adaptive DBS or AI DBS, where the onboard system is learning about the brain physiology. It can differentiate different behavioral states. It can assess changes in physiology over time. It might as an input function, have medication dosage, and maybe it could recommend its own settings or even medication adjustments or timing. And Julia Newman wrote a nice review paper on this in Neurotherapeutics a couple of years ago. I do think that this evolution has a potential to be informed by learning from implanting DBS in different diseases, not just movement disorders, and can also learn from our experiences in epilepsy and across devices. So this is one reason why functional neurosurgery is so exciting, is this ability to think about how the brain works and how we can modulate its activity as we understand the pathophysiology. So let's review the current state of the art. So now this has been several months now since this device was approved in Europe. So this is the sensing-enabled device from Medtronic. This evolved from the PC plus S. The plus S stands for sensing. And now this has been available in the US for now for several months, maybe a year now in Europe. Time flies in the pandemic. So we can stream some activity while the patient's in clinic. There's very limited ability to record from the device when the patient's out and about, at least with the current features that are unlocked. Having said that, this device is moving towards use as a closed loop adaptive system. There is now a clinical trial that's open and enrolling. We're participating in this at MGH. Dr. Harrington and our group has recruited a couple patients into the screening process. And so I thought I would just share a couple things about that. So this is a historic clinical trial. To be clear, it's the first full clinical trial of adaptive DBS. And let me just say, there have been several reports of how to do and how to think about adaptive DBS in the setting of externalized DBS leads. And I'll specifically review some of the very recent publications on using implanted devices, the initial attempts with implanted devices to do closed loop DBS that are outside of this trial, really go beyond this trial. But this is a current state of clinical trials. So this is it's a prospective single-blind randomized crossover design. It's multi-center. I think 6 sites in the US and 6 in Europe, roughly. 36 patients, total evaluation period 15 months. The primary endpoint is a comparison of standard continuous DBS, duty cycle, to adaptive DBS by counting hours of on time without troublesome dyskinesia, as reported by patient diary. So subjects are going to receive the standard continuous DBS at baseline to just establish the expected clinical efficacy. Then they'll be randomized and there'll be a blinded evaluation in comparison of 2 different adaptive algorithms. So I'm just going to show you a plot from each of the kind of seminal works. And this is work from the Oxford group that illustrates the single threshold mode. And the trigger here in Parkinson's disease, the initial trigger has been beta activity, which in not all patients, but in many patients with Parkinson's disease is elevated and corresponds most often to the symptom of bradykinesia. So there's a pathologic elevation of beta oscillatory activity in the basal ganglia. So the idea here, so here's the LFP signal. Here it's filtered to see the beta burst. This straight line here would be the threshold of beta power or amplitude. And this squiggly line is showing you the average, essentially, amplitude of beta power. And you can see when it crosses the line, you go up, trigger is turned on. That's this square wave here. And then here's a schematic of the stimulation. It ramps up, it's on. Stimulation is on. We know that it can drive the beta power down. That's the correlate of symptom improvement. And you see the beta power dip here. Stimulation goes off. Trigger is kind of reset. And here the beta power increases, and you can see this continues. So this is a single threshold mode. There's just one line. Cross it in one direction, the simulation goes on. Cross it in the other direction, it goes off. The work of Helen Bronte-Stewart at Stanford has taken a little bit different approach, where this is a dual threshold mode. And there the idea is that there is a kind of normal range of beta activity. Let's get that back. And if beta activity increases over this range, DBS is turned on. And if it decreases, it's turned off. And the idea here is to try not to undertreat and try not to overtreat. And you can see an example here on the right, which is showing a beta power going down as DBS voltage goes up, and limb, this is accelerometer data, is increasing, essentially patients moving better. Okay, so those are the 2 modes that will be tested in this clinical trials. This is very exciting. and hopefully will lead to FDA and CE approval of the technology that already exists on the device and it can be unlocked. And that'll be really another just huge milestone in the field of DBS. Okay, so there are also ongoing what we call investigator-initiated studies where people in the United States through the BRAIN Initiative and other mechanisms are using devices on their own to start figuring out closed-loop DBS. Okay, so here I've chosen 3 examples from the recent literature to review that those who are interested in this field should really be aware of. So the first that we'll talk about is from the Oxford group, Peter Brown's group. And this is, this strategy in this report was developed for essential tremor with the signal being derived from the DBS electrode itself and the BIM nucleus. So one thing to note is that this processing is not done on the device. Leads are externalized. There's a computer where the algorithms are run. It's connected to simulation. And so this is done in the setting of having leads externalized before stage 2 DBS, which gives a great opportunity to test these paradigms. So what this group did was to develop actually 2 algorithms. So people with essential tremor some people will have a tremor really every time they go to initiate a movement. So you can basically make an assumption if they're going to move, that their tremor is going to come. And so that's what the top plot is showing, where the algorithm decodes when this movement is happening. And it does that through assessing the spectral activity. But there can be another type of tremor, which comes from, which can be postural tremor. So the patient is in a certain position, they're not moving, they're holding that position, or maybe they're doing some fine motor something, and the tremor is very active in that particular position. So they develop a different algorithm to detect that, and you can see that here. Here's the EMG, and here's the signal essentially. Here's an average of the tremor power, and here's the signal turning off and on. Okay, here's a different approach for a few reasons. So also an essential tremor, but this is University of Florida group, where they have ECOG strip electrodes implanted over motor cortex, and in particular over hand motor cortex. The idea here is to, again, an external device is used to do training of the algorithms that will determine the detection parameters. And the patient comes in after being implanted and goes through different training periods and specific tasks. And here's an example of just a schematic of what that looks like. And here's streamed data from motor cortex, contralateral movement. You can see beta desynchronization. Here's ipsilateral hand movement. What else can I show here? And these are they reported on 3 patients. And this is a further illustration of what the data looks like. Now we're hearing data, now we're seeing data from the thalamus itself. Contralateral hand movement here. You see the beta desynchronization. You can see in the thalamus, it's not as strong. And here again, you can see it kind of zoomed in, movement onset. Detected in the thalamus, but obviously much stronger in the cortex. And this is an estimate of when stimulation will be applied. So they train their algorithms and then they can implement this. And this is an example of what this looks like. Take-home message here is that it makes a lot more sense for this type of approach where you want to stimulate whenever the patient's going to move by recording from the cortex. And that symbol is very robust and avoids problems of stimulation artifact in the DBS lead, avoids problems of where you have to set the bipolar recording pairs, for instance, which can affect where you might stimulate. And DBS for essential tremor can work extremely well when you're stimulating the right spot. And if you have to move that spot too much, then the efficacy is not as great. So the idea of decoding the intent to move from the cortex and then stimulating the thalamus makes a lot of sense. And in fact, they showed very nice results in this initial paper. And that, of course, was with the implanted device and cortical leads. So you can see the Oxford group also reported in their kind of acute study tremor reduction that was consistent with regular standard DBS. And there was an energy savings, importantly, because just less current is being delivered. So this has advantages for not having to have the battery change as much, which is very attractive to patients. And even if there was a rechargeable, let's say, not having to recharge it as much. So DBS is supposed to be a quality of life improving surgery. So anytime we can do something like that, that essentially lowers the burden on the patient and these chronic diseases where they're going to have a brain device implanted, I think is extremely useful. And the University of Florida group showed a similar result. Good tremor control consistent with what we expect from standard DBS, but with the cost savings on the energy side. Okay, and here's a really exciting result that is recent from the UCSF group, where these are patients implanted with a version of the device that can stream information wirelessly to a tablet. And these patients even had data kind of coordinated from an accelerometer on a watch that can give a readout of tremor, bradykinesia. And the group was able to train algorithms that use this information that was collected in patients' homes to adjust the settings on the device. And this is just an example. And again, using cortical and subcortical recordings. So here, these are, I think, this is one patient example. 3 canonical frequency bands that are generally associated with tremor in the alpha band, beta with bradykinesia, and dyskinesia with gamma oscillatory or activity. Actually, these are 5 subjects, because the point of this is the heterogeneity between subjects. I think this is subjects, now I've confused myself. Or contact locations, things, subjects. Because you can see that the activity, for instance, recorded in the STN. So here's strong beta activity, this particular patient. Is not necessarily present in the other patients. And so here you can really get a sense of the need for personalization in a closed loop system. DBS isn't a one-size-fits-all approach, and even closed loop DBS isn't a one-size-fits-all approach. It's important to really be thoughtful about this. And so they were able to develop a control signal from beta, from subthalamic beta activity, as well as a different control signal potentially related to dyskinesia from cortical gamma activity. And here you can see adaptive DBS working better than open loop. So very promising and really very exciting to have data collected in a patient's home that's used to inform the closed loop system. We're really starting to see some very exciting advances in this field. Okay, so let's see. Why did I put this slide back? Aha, because, okay. what about getting to this stage where the device is, in the example we showed, the device is not doing all that work itself in terms of incorporating the data collected at home. So how do we get to a stage where that's possible? One is the application of machine learning techniques, which currently have to be done offline. There isn't the onboarding processing ability to do these really kind of high level decoding problems on the device. Here's an example from a review paper that was written by Timon, Merrick, and Victoria Peterson, Julian Newman, with a schematic that shows the type of processing features that we would like to have on the device at some point. For the time being, this work has to be done separately. And our own approach to this has been to use the operating room to record this data, because we don't have patients implanted with the PC+S or the RC+S device, but we do intraoperative experiments with cortical strip electrodes. So this is now an old data set, but one that we've continued to look at, now apply machine learning techniques to in terms of decoding movement. And we wanted to ask the simple question of whether we could decode this more easily in the cortex and the STN, and whether it was helpful to have the STN recordings. In this initial data set and approach, the answer was it's easier to decode from the cortex, and the STN is not particularly helpful. This is a schematic that just shows you the steps involved in setting up a decoding pipeline to answer that one question that we ask here, which is very simple, which is when's movement onset occurring? So this is a lot of work, but these are important steps for thinking about how this evolves in the future. And as kind of a transition to the next part of the talk, I'll show you this, which was a correlation that Timon and Julian did to determine whether there was anything predictive in functional or anatomic connectivity that would help to predict which locations gave the best decoding performance in terms of movement onset. And not surprisingly, when looking at fiber tracking, at tractography from a kind of global connectomic database, leads that were in primary, or contacts that were in primary motor cortex, gave the best coding performance. So this is kind of validation that this technique is giving us the expected answer. Interestingly, when you look at the fMRI-derived functional connectomics, you see the expected hotspots for electrodes that gave the best decoding ability as predicted by the functional connectivity, but also areas in the cerebellum showing up. So this is very interesting because it's a technique, a way of thinking about the data that may help us understand the brain networks involved. And this is essentially preliminary in the sense that this is a few patients we haven't specified the connectivity related to specific symptoms, et cetera. But we do think this is a very interesting approach to thinking about, for instance, where's the best place to put a cortical sensor? And that may be obvious with this question, where's the best place to decode hand movement? It may be less obvious for something like a speech symptom. And may need even more help for cognitive side effects of DBS. If, for instance, we move into an era of also doing DBS for non-motor symptoms of Parkinson's disease in conjunction with treating the motor symptoms. On that note, speaking of non-motor symptomatology, I'll show you these two examples of an approach which is very similar in two separate groups to use stereo EEG to guide DBS. And now we're talking about for major depressive disorder. So on the left is a schematic of the approach from the group at Baylor and UCLA to implant regions of interest that are implicated in network dysfunction and depression with DBS leads in presumed target of interest, but also recording from other areas that would be expected to provide useful information for how to do DBS programming. And this approach uses the segmented lead in the Boston Scientific System. And then the group at UCSF has taken a very similar approach in terms of the hypothesized areas for implantation to map depression network and responses to stimulation. And these patients after SEEG subsequently implanted with RNS. So I think the outcomes of these trials alone are going to be very interesting, as will be the outcomes related to different device and approaches used. So I think this is very, very exciting, and we're just now starting to see the first reports of any of these patients. It's very difficult to do this work. And so I think both these groups are really to be commended for advancing the field in terms of how we think about treating these disorders. And I thought it was important to note that that approach didn't start there. I think this is kind of often overlooked strategy that's been published for pediatric dystonia. By Dr. Sanger, I think primarily leading this. And this is very interesting, where multiple targets in the basal ganglia and thalamus are implanted with SEEG leads, including Benke-Fried electrodes with extended microwires, where single unit activity is recorded. And looking at the spike data and response to simulation is actually part of the algorithm for figuring out whether or not to implant the patient. You can see that here on the schematic. So I think we're going to see this approach more often for different diseases. And I think what we've learned from doing stereo EEG and epilepsy, and if you were in the last lecture, I'm sorry, with the technical snafu there, I think I lost some slides on stereo EEG that weren't saved because we didn't spend a lot of time talking about the stereo EEG data itself and how to use the semiology to understand the network. But what we have learned from using this so much in epilepsy is that it's safe. And for these diseases, major depressive disorder and others for which patients have just failed over and over again medical treatment, I think it's perfectly reasonable to invasively assess the network to figure out the best place to potentially perform neuromodulation. Okay. Let's shift into this area, which is frankly a new area for me, but been very motivated by the fact that Andy Horn, the creator of Lead DBS, is moving to Boston. He's going to work with Mike Fox at the Brigham and also have a presence at MGH, working with some of our neurosurgical data. And Andy and Mike published this very nice review paper in Neuroimage, and I've taken a few of their figures to show the potential of this type of approach. So one thing to just recognize is that through patient-specific imaging, whether it's functional or structural, and so when we think of connectomics, we can think of those essentially two modalities: tractography and functional MRI, typically resting state, to derive a connectivity map in an individual person's brain. However, it's really difficult to do that accurately on a single patient, certainly with a single scan. And so normative connectomes are potentially a way around this problem, or at least a different way to think about this as techniques are developed to improve single patient connectomic imaging. Because combining patient data obviously can be more powerful, can bring out these relationships. And so this has been applied now in relation to DBS for Parkinson's disease, essential tremor, OCD, to show that regions that are most often associated with clinical improvement in DBS and these various disorders are associated with specific network topologies. And so, okay, so here's an example of a functional network. And this is something similar with the structural network. And at the top is OCD, showing fibers engaged in patients who respond that are red, and the fibers predominantly engaged in patients who don't respond in blue. And the bottom is Parkinson's disease, showing that the activation of these premotor fibers being important in patients who responded. And so I think the take-home message here is we're starting to define connectivity targets that we might like to engage when we do DBS. And maybe we'd like to have confirmation that we're doing that. Or not. Maybe we don't need to. Maybe we can map this out with imaging before surgery and just put the electrode in the right spot and make the assumption the network's going to be activated. And I think that's a very interesting question for future work. And then I just pulled, there were 8 kind of lessons about opportunities from this review paper, which I'll show here. Explaining and predicting clinical improvement is a good one. I think the flip side of that's also very important. Explaining clinical failures, linking different DBS targets to the same network. Example, depression in OCD potentially. Defining networks for specific symptoms, doing more to improve patient-specific connectomes. Of course, mapping side effect networks. And this is what I just mentioned, guiding the surgery itself. And of course, programming. Potentially using multimodal therapy, so non-invasive brain stimulation to target one aspect of the network. And of course, this one, critically important, there's the potential to understand things about functional and structural connectivity that explain aspects of disease or treatment that weren't recognized without doing that. So, one question is, if we can do all of this fancy imaging and determine where the networks are that need to be modulated, and we can do DBS asleep and an MRI, and we can do that all at the same time, why do we need to do awake DBS surgery? So I think this is an open question. I'd say short answer is patient specific variability in physiology is one reason. We don't know who's going to have a good beta signal or who's not unless we record. And someone who has that present in the operating room isn't always going to have that to the same extent outside of the operating room. And then there's the ability to understand different symptoms and different behaviors, and placing cortical sensors in regions that are important for specific patient symptom control or side effect avoidance. And in my group, we've been looking at the role of basal ganglia and thalamus in speech production. And so this is an example of an intraoperative setup to do that, where we put a strip electrode over speech motor and auditory cortex, while we have electrodes recording single units and local field potentials in the subcortical target as the patient is producing speech. And this just, the middle image just shows you the microelectrode holder, microdrive on the rest of the robotic arm, and you can see the wires coming out of the burr hole here from the temporarily placed ECOG strip electrode. But you could imagine if a patient has a particular problem with speech, bradykinetic speech, stuttering speech, as a primary component of their Parkinson's disease, that we might place a cortical sensor in a speech-related area, in addition to a sensor that maybe is in their hand motor area to help with their hand tremor, their bradykinesia there. So I think there's a lot of potential here, and awake DBS surgery, I think, is alive and well to help answer these questions about the relationship of the structural and functional imaging to the actual physiology that we can record during awake behavior. And those questions can't all be answered with the implanted devices because, at least for the time being, the areas in which they can assess are greatly limited compared to what we can do in the setting of awake DBS surgery, even though there our main constraint, of course, is time and patient fatigue and participation. Okay, and that's all I have there. I'm happy to take any questions, discuss anything in the presentation, or anything I didn't discuss.
Great. Thank you very much, Dr. Richardson. It was an excellent lecture. Very complete, and give us the current vision of DBS, ADBS, and intelligent ADBS. Excellent. It was really nice. Yes, we have some questions right here. Ali Bayramov is asking, how effective is deep brain stimulation for refractory obsessive compulsive disorder? How does it work, and in which mechanism?
These are all great questions. The one randomized controlled trial that attempted to show that this was efficacious was not able to do that. But in our experience, and certainly experience at MGH, where there have been a lot of patients implanted in the nucleus accumbens ventral striatum, that the efficacy on average is quite good. And so when we do this, we really expect it to work when we have the proper patient selected. The target is really the anterior limb of the internal capsule. At least that's what seems to be clear from connectomic work and just the evolution of the targeting. How it works is not clear. We had one patient recently, this is anecdotally, who is almost a complete responder to DBS. Why that is and exactly what circuits are being modulated, is not clear. And I think one of the reasons for that is the number of patients, the criteria for implanting someone with OCD with DBS is very high. The Y-box score that's required is high. So these are people with very severe OCD. Many of these patients, it is extremely difficult for them to manage to get into the hospital or to finish the required other treatment attempts that they need to do. So one problem that the field suffers from is not that many cases of OCD DBS being completed. So that doesn't answer the question. Short answer is I don't know why it works beyond the basic stuff that that's been reported about network modulation, and but I have seen it work quite dramatically. So it's clear there's something there.
Yes, right. We have another question. In your practice, Dr. Richardson, have you considered DBS and connectomics for treatment in eating disorders, such as obesity?
In my own practice, I haven't done this, and there are two examples of well, at least in the U.S. In China, there have been some studies related to this. I'm not familiar with those, and in Europe, I'm sure there's some trials I'm not familiar with. In the United States, Don Whiting at Allegheny General has published a couple times in his case series of hypothalamic stimulation. My understanding has been hard to get a persistent response for overeating, eating disorder. I did see promising data presented by Casey Halpern at Stanford related to using the RNS system to detect a, let's call it overexuberant reward signal related to binge eating, and potentially to modulate that with RNS in the nucleus accumbens. And that was very preliminary data, but seemed promising. And that's the state of the art that I'm aware of related to at least overeating disorder.
Okay, and there's another question quite similar in its pathophysiology, and it's about DBS in order to treat drug-related addictions. What's the role that if there's
drug addiction?
Yes, if there's any role of DBS in drug addictions.
Yes. So over the years, there have been various studies, and I don't honestly know what we can presume that that there hasn't been a blockbuster there, because if I looked this up several years ago and kind of did a survey of the literature. So one thing that's really hard about addiction is you have to, I guess you don't have to, but one typically picks the type of addiction: alcohol addiction, or it's opiate addiction, some other drug-specific addiction. And these patient populations can be very difficult to work with. I'm actually surprised that, and maybe there is a trial or some a project ongoing that I'm not aware of, but I do think that we're going to see this. I think this is an obvious place for DBS to go. So I think there's definitely potential there for drug addiction as we understand these circuits better. And for instance, OCD and movement disorders. I mean, the basic answer to the question before about OCD is, I think we can make an assumption that the effect is similar to what we see in movement disorders. It's just that we don't know exactly what the signal is, the pathologic signal that's being interrupted in OCD. But there's some sort of pathological probably synchrony between brain regions that doesn't let normal behavior proceed and that is somehow blocked by DBS.
Okay, great, thank you. Dr. Madriñan asks, do you perform magnetic resonance guided focus ultrasound? What's the future for this type of therapy in the short term?
Yeah, I don't. So I'm not really qualified to answer that. We don't have a focus ultrasound at MGH yet. My understanding is that that the morbidity associated with this for lesioning the thalamus, for instance, in essential tremor, has improved significantly. And I think initially when focus ultrasound was kind of getting going, at least my read on the literature was that the associated morbidity was too high in relation to DBS. And so I think that's changed now. My personal opinion is that focus ultrasound is not a good option for most patients with Parkinson's disease because they don't have just one symptom. They have more than one, and they have, I think, a greater need for a therapy that can be modulated. As focus ultrasound evolves, could it replace laser ablation, let's say for treating epilepsy? I think that's reasonable, but I don't have direct experience with focus ultrasound.
Okay, and also Dr. Madriñan asks, do you think magnetic resonance guided focus ultrasound might have a role for fenestration for the floor of the third ventricle for obstructive hydrocephalus?
That is interesting. Short answer is I don't know. My understanding is that the physics of focused ultrasound have not advanced to the point where that could be safely done. But honestly, I'm not sure.
Okay, and Dr. Sanchez is asking, what's the future for DBS for depression and Alzheimer's disease? Well, I think you talked some about this in the lecture, but if you can complement anything.
Sure. I think there is still, I'm a strong believer in the potential of DBS to help patients with major depressive disorder. And I think, the work that Helen Mayberg has done at each institution where she's practiced has, I believe the results, that those groups have gotten. And they have a very thoughtful approach to using connectivity prospectively. And have a very good sense of patient selection. It is hard to translate that experience in general. It's highly specialized, but I think, frankly, we should see more attempts to recapitulate the basic work that Dr. Mayberg has done. I think these stereo EEG attempts are going to advance our understanding dramatically, and we'll see how long it takes to really have a kind of order of magnitude leap forward, which is what we need there. Alzheimer's disease, I'm less hopeful about. I don't understand the mechanism through which DBS could help patients with Alzheimer's, and I'm a little skeptical in general of that approach, but it could be that I just don't have an adequate understanding.
Okay. Dr. Richardson, in your practice, do you favor an very early DBS surgery for patients with Parkinson's disease? Or what's your opinion about that?
Yeah, everyone's version of early and very early is different. So I would say, in general, yes, I favor early intervention. It's really, again, about quality of life for the patient. But as soon as someone is having a problem with adequate control of motor symptoms with medication, or they're having motor fluctuation, or some side effect or some inability to advance their medication to control their symptoms, and they clearly have Parkinson's disease, then as soon as they're ready, it makes sense to do it. I think there are a lot of benefits to doing it. As soon as the patient's ready, once they cross the threshold into candidacy, then yes, I'm an advocate for that for sure.
Okay, thank you. In your experience, what do you prefer for subthalamic nucleus between intraoperative MRI versus microelectrode recording guidance?
Right. So this is another good question. I am biased for both. And so the short answer is I don't favor one or the other, and I try to leave this up to the patients. And so this is also the way the residents ask this question too, when they rotate onto the service. How do you decide, or how's the patient decide? In our practice, it's pretty easy. I explain what our biases are, and I have a bias to wanting to provide a sleep DBS, at a very high level and maintain a good experience with that, because there are patients who will not have awake DBS. And so it's important that we do a lot of a sleep DBS so that we stay good at it and help to develop that technique, which is very helpful to some patients. On the other hand, I like doing awake surgery. We have NIH-funded research to explore different behaviors related to basal ganglia and cortical activity, and I tell patients that, and patients tend to fall into categories of being very interested in that and kind of interested in the idea of being awake, or they fall into the category that they're definitely not interested and would like to be asleep. And then sometimes you have a middle ground of patients who they see the value of being awake. They get the sense that our neurologist, who is fantastic and will be in the operating room there, that they would get to work with him, and they prefer that. But maybe they don't want to, not so interested in the research, which is fine. So I'd say those are general categories. In Pittsburgh, we published the first 20 STN cases that we did in the Magnet, with those that we did with microelectrode guidance. And like other groups who have done something similar, we couldn't see a difference in outcomes or side effect profile was one thing we looked at in that paper, or complications. So that was just open label, single center experience, but that experience matched that of others. So I think short answer is efficacy should be the same. It's important to be honest with the patient, what your motivations are for either approach, and the patient picks.
Excellent. There's another question. Do you favor STN versus IGP DBS for Parkinson's disease, or what's your preference?
I don't favor one over the other. For instance, I've seen GPI produce a great tremor control result, where typically it's thought that the STN is the better target for that. We do tend to implant GPI in patients who have dystonia as a primary component of their Parkinson's disease, even though the STN is also a dystonia target. We have taken that kind of traditional approach. For patients with dyskinesia, that can be sometimes more difficult question, depending on what we think the cause of the dyskinesia is. And if it's medication, there's a real need to lower the amount of medication to control the tremor, and we think that would be most helpful. Then we might pick the STN, even if there's a chance for causing some temporary worsening of dyskinesia that has to be worked around. So it does seem that in the STN you can lower medication more easily after DBS. And there's the older population who, if there is any concern on the neuropsychology, we tend to implant the GPI, even though the literature does not necessarily support that there's a difference there. I think it is a larger target, so maybe on average you have less chance of maybe a misplaced lead in a thinner older person's STN, and therefore maybe that's a reason that it's you might have less chance of a cognitive side effect. But for the most part, we don't always favor one or the other. We try to tailor it to the patient.
Yes. The next question is, what are the most frequent complications encountered during DBS surgery?
Well, let me think about that. Well, I think the one that is, how about it this way? The one that I'm most concerned about is not having the electrode in the intended target. And so in our awake cases, we get a CT during that admission, and we compare it to the surgical plan, because there are some cases where someone's made a mistake on snapping the block on and the lead's moved or something like that. And I prefer to fix those as soon as they're detected, as opposed to discovering it later. Another philosophy is to wait for any brain shift to settle and get a post-op CT a month later. I would just hate to discover a basic problem that far down the line, and we'd like to have the feedback that we've put the lead in the spot where we expect it. And then for a sleep, I do think the MRI, even though a sleep CT guided verification has a really high accuracy. My preference is obviously to do those sleep cases in the MRI. The worst complication, I mean, the one that you should really watch out for is, I think, venous infarct. Aside from just being very careful with the planning and avoiding vasculature, one thing to watch out for is a venous infarct, where the case can be done very well, but maybe there was some brain sag and the patient was dehydrated, and there's an unexpected venous infarct that shows up, and patients can recover from that pretty well, but you want to avoid it if at all possible. So you be very careful in opening the dura and ask the anesthesiologist to make sure the patient's hydrated during the case. Is another trick.
Thank you, Dr. Richardson. And the last question for this session is from Dr. Sanchez. During the assessment of choosing the ideal target for DBS positioning in Parkinson's disease, can we consider the local field potentials to be similar network as occurs in the epileptogenic zone in epilepsy?
Yes, so we do. We can do that, and there's certainly publications that support doing that. Primarily from Haggai Bergman's work and other publications that have used, for instance, the Haggai component on the neuroomega system, which assesses beta activity, can help with placement. So short answer is yes, I think we should be doing that. Longer answer is we don't fully understand the variability patient to patient and why we, for instance, don't see as much beta in some cases, and what other biomarkers we should be looking for intraoperatively. I don't think we're at the stage of movement disorders where everyone needs to get SEG, to assess this. But I think there are probably some examples of difficult cases of, for instance, patients who would be expected to respond to STN or GPI stimulation, and for reasons that are difficult to understand, just don't. Maybe those are patients that need broader simulation of the network.
Thank you very much, Dr. Richardson. On behalf of CIEN, I would like to thank you again, Dr. Richardson. We are really grateful for your participation and for your time in the 2021 IW BNC.
Dr. Pena, thank you. Thanks to everyone for the invitation. Appreciate it.
Thank you very much. For all the audience, keep in mind this lecture will be available on our website starting from the next week. We hope all the audience to be connected for the next conference, which is Modern School-Based Surgery, 6+1 School-Based Projects by Juan Fernandez Miranda. Please follow the link pinned on the chat screen and check the program schedule on our website, seeingwhos.com. Thank you very much.
All right. Great to see you. Thank you.
Thank you, sir. Thank you very much.
Okay, yeah, and do let me know anything I can do to help once you get started again down there.
Yes, of course. Thanks you a lot for that offer. It's an honor for us and for the group.
Okay, great. I'm sure Juan will give a great lecture.
Yes, you will know soon, Dr. Paulo Sanchez, who is going to be doing an observership with you.
That's right.
Soon. Yes. Yep. So hopefully everything's going to be wonderful.
Oh yeah, I think it will be. Thanks for sending him. So that'll be good.
Thanks a lot, Dr. Richardson,
right.
And goodbye.
Bye-bye.